So let's go ahead and go on to the next topic here. So part 2 of our webinar today is going to be on conjugation. And conjugation is a different form of genetic engineering. So again, I'm Dr. Dennis Mishler. I'm at the University of Texas at Austin, and I've been part of iGEM for a number of years now. Okay. So conjugation is a naturally occurring process. So bacteria in nature, they can naturally transfer their DNA from one to another horizontally. And the way this happens is if one bacteria over here contains a plasmid that encodes for this pilus and the machinery to potentially make contact with another cell and then initiate a transfer of its plasmid material into the other cell. So this is the donor cell on the left, and this donor cell contains a plasmid with the pilus, and the pilus then interacts with a recipient cell, which does not yet contain the plasmid. The plasmid, once these two cells are connected, then starts to make a copy of itself and sends that copy over to the recipient cell. And by the time the process is done, your former recipient cell is now a new donor cell. And both cells contain a copy of the plasmid, and both cells are now able to further transfer that plasmid to other cells. So that's the naturally occurring process of conjugation. But from a genetic engineering perspective, what we want to do is we want to use this process for engineering bacteria that don't already have easy to use transformation procedures. Okay. And there are a bunch of bacteria that this applies to, particularly non-model organisms. Organisms that are fairly recently discovered or just now being worked with, that nobody has figured out how best to transform them. People will often try to first use conjugation because it just tends to work with cells. Okay. Conjugations will not work with all organisms, and I do not promise that you will have good results. This is just another option. To do a conjugation, though, you're going to need a specialized donor strain as well as a plasmid that has a specific origin of transfer. Those are the two special things that you don't normally think about when you're doing transformations. Okay. And an origin of transfer is different from an origin of replication. Okay. So you need your strain of donor cells. So these are cells that are normally oxytrophic for a particular molecule. And what this means is that if it's oxytrophic, you have to provide that organism with that specific molecule. If you don't give them that molecule, the bacteria won't grow. So your donor cells need to be oxytrophic. The donor cells should also probably contain all of the transfer machinery within their own genome, within their own chromosome. And this is different from nature. In nature, all that transfer machinery is going to be encoded on the plasmid to allow for the transfer. But when we're genetically engineering and making things, we'd rather that machinery be on the chromosome so that our plasmid can be as small and contained as possible. Separately, as I mentioned before, separately, these donor cells will also need to contain your plasmid. So you'll need to first transform your plasmid into your donor cells, maybe using the procedures we described in part 1 of today's webinar. But you're going to first need to make that donor cell contain your plasmid before it can transfer your plasmid to a recipient cell. Okay. Additionally, just a heads up on the procedure, you're going to need quite a bit more time to carry out this procedure. So transformation can be done pretty quickly. A conjugation may take several days. All right. You're going to need to make sure that your plasmid is compatible for conjugation. So your plasmid needs to have a distinct origin of transfer as well as an origin of replication. Okay. So all plasmids need origins of replication, but only those that you're conjugating need a transfer or origin of transfer. Okay. And importantly, you may have to try multiple experimental conditions to be successful. Commonly, people will not just do one conjugation at a time. They'll do like five. So here, this is a blood agar plate, and you can see there's 5 different conjugations, 1, 2, 3, 4, 5 being done on this plate, each one with different ratio of cells. So to make sure the ratio of donors to recipients is appropriate for these organisms. Okay. So there's a lot of different things to think about and try on conjugations, and we're going to try to walk through a little bit of this, but it's not really that difficult once you start doing it. So here's some basic terminology just so everybody's on the same page as you go forward. So first off, I want to highlight that all of this conjugation information I'm using on this slide, the next slide, this all comes from this procedure linked right here. So if you need to, you can go to this page. This PDF will be online later this week. You can go to that page and you can read all about the procedure and some of the details. So first, donor cells. I mentioned this earlier. They have to be oxytrophic, and specifically for the procedures I use, are oxytrophic. They're oxytrophic for DAP. Okay, these cells will only grow if you give them DAP. If they don't get DAP, they don't grow. So that's great. All right. DAP is diaminopimelic acid. It's a very tough word to say, but this molecule is important for cell wall formation. So if you don't give them DAP, they shouldn't be able to grow cell walls, and thus they shouldn't be able to grow. Okay. Origin of transfer, also known as ORET. This is the part of the plasmid that initiates the transfer of the plasmid into your recipient cells. Okay. Not all plasmids have an ORET, so you'll need to check that. If you're trying to do conjugation, make sure you have one of those. All right. Recipient cells, those are simply the cells that you wish to add your plasmid to. So, it's just whatever your organism is that you want to work with. Sometimes we'll use this terminology, transconjugates. This means the recipient cells that have potentially received your plasmid. So basically they're the recipient cells that have been hopefully conjugated. And when you're doing a conjugation, you're going to need two different types of plates. Okay, you're going to need a non-selective plate that you use with a mixture of cells. And this plate will have both donor and recipient cells on it that grow together. All right, and that was a plate I showed you on the previous slide. So over here, this is a non-selective plate with both donor and recipient cells, allowing them to grow together to potentially conjugate. You're also going to need a selective plate. And these selective plates should only allow the transconjugates to grow. All right, these plates will not have DAP, and they will have antibiotic presence. Okay, so these selective plates will kill the donor cells, hopefully, and they will kill or prevent the growth of the non-transformed, non-conjugated recipient cells. Sorry, my throat's getting a little dry. Okay, so let's talk about the two real steps in conjugation. So this really comes down to two steps. There's a little bit of prep time for each step, but there's really just two. The first step, step one right here, you got to mix the donor and recipient cells and place them on the non-selective agar plate with DAP. In this condition, what happens is that the donor and recipients, as they're growing next to each other, hopefully make contact and exchange the DNA. Since there's DAP present, the donor cells can grow, and since there's no antibiotic present, the recipient cells, with or without the plasmid, can also grow. This will need to grow for some period of time. Sometimes it's only a couple hours, but often it's multiple days. So I've had people do conjugations where they wait 2 or 3 days, and I'm sure there's longer procedures as well. But you have to let some time pass, okay? And this growth has to occur at whatever temperature is ideal for your organism. So if your organism normally grows at 25, you need to do this incubation at 25 degrees Celsius, all right? Once this process is done, you move on to step 2, and this is the data related to step 2 here. In step 2, what you're going to do is you're going to scrape off the mixture of your cells. So these cells here are going to be scraped off, and they're going to be placed onto another plate. And this new plate is going to be a selective plate. It's going to contain antibiotic, but no DAP. And as I mentioned before, it's on the right-hand side, the top 2 plates. The top plate is basically day 1 or at the very beginning of that process, and then this bottom plate is a day or 2 later, once the cells have been growing on that new plate. Okay. Because there's no DAP, the donor cells shouldn't be growing, and they should be hopefully dying off a little bit. And because there is antibiotic, only the recipient cells that have the plasmid should grow. However, I would never recommend going straight from this first selective plate to assuming that you have been successful. I would always recommend, and I say when needed, but I would always recommend it. I would always recommend taking from that first selective plate and then streaking out those cells on a second or even a third selective plate, okay? And the reason for this is that you want to make sure you're not accidentally carrying over any donor cells or any false positive cells of any sort, okay? So what you can do is you can take a little bit from here, And you can either streak it out if you have a certain bacteria that grows in little colonies, you can streak out. Or in our case, we have a different type of bacteria we're working with here, which grows differently, so we couldn't do that. Or you can just take a little bit from the sample and put it onto a new fresh plate. And this new selective plate, you should get rid of all the donor cells, and you should get only positive results on this plate. Okay. And so here, this is some unpublished data. This was a conjugation reaction for using the organism Oscillatoria lutea, which is a cyanobacteria. And what my students have conjugated here are not actually the Oscillatoria, but instead the symbionts that live with the Oscillatoria. So what they did is they conjugated from E. coli into these other bacteria that live with Oscillatoria. The Oscillatoria grew out, and you can see the symbionts are now fluorescent, so they've been conjugated successfully. Okay, but this conjugation procedure here, what my students did, this probably took them about a week to conjugate. Okay, but none of our transformation procedures work, so that was the only option they had. Transformations didn't work. We got to conjugate. It takes longer, but hey, we got a good result. Okay, so conjugation is often best to confirm that your recipient strain has been modified by doing 16S sequencing as well as plasmid isolation and sequence confirmation. Sometimes you'll get residual donor cells hanging out, and if you can't easily tell the difference in phenotype between the donor and recipient cells, you really need to do some sequencing to confirm that you have the right cell present, in addition to confirming your plasmid is correctly made. Okay. I highly recommend starting with this Barrick Lab conjugation procedure page. That's the one my students always use. That's what we use. And you can modify that procedure as works best for you. It's always fine in science to modify a procedure as long as you write down those modifications and you keep track of it. It's really important, though, to note that other procedures will also work. If you're working with somebody who uses a different procedure, use their procedure. This applies to any experiment. If somebody you're working with is an advisor and a mentor, if they use a different procedure, use theirs unless theirs is not working. They're going to be able to help you a lot more than, I can help you with using my procedure. Okay. So if you talk to me about how to work with this procedure, I'll tell you what I would do, but your reagents may be different. You might be doing a slightly different experiment or have a different type of DNA sequence, and it may be difficult for me to help you troubleshoot that procedure. Okay. So I'm always happy though to talk or give suggestions, guys. Don't be afraid to reach out to me. Importantly, conjugation can take multiple trials to perfect. And one important thing is that you can do multiple ratios or incubation times during the same experiment, right? On a plate, you can put multiple reactions down there and try several things at once. You could always do 2 plates, let one incubate for half a day, another one incubate for a day or 2, and then move on to the next steps. So you can do multiple at the same time. Okay. And this is just a summary again of how the process works. So if you're really confused about things, this is really the take-home message here. The DAP oxytrophy is really important, and the fact that you have a selectable marker on your plasmid is really important, and that's what allows you to get through this whole process. Okay. So hopefully you guys learned a little bit about conjugations and some of the steps in this procedure. There are, again, a lot of stuff online that you can look at and take and work with. I had a link on the previous slide that showed you one procedure. There is this short little YouTube video that shows you an animation of sort of the naturally occurring conjugation, what happens there between 2 cells. So you want to look at that. We're going to go on to sequencing in a little bit, but I'll pause again for questions right now. So thank you guys for listening to this part of the webinar. If you have any questions, please write them in the chat or in the Q&A. Either way is fine. I'm happy to take any and all questions right now. Okay. And we already have one question, so I'll go ahead and answer that one while people are maybe thinking about other questions. So the first one is, one second here. What do I mean by wash? Yeah, so by wash, what I mean is when you're taking cells from the first plate, and before you put them on the second plate during the conjugation procedure, you'll actually mix them with like some buffer first. Make sure they're mixed, centrifuge them down, remove the buffer. You might do that a second time. And what you're doing is you're just trying to wash away any chemicals, any buffers, anything like that that might have been from that first plate, so it doesn't come over to the second plate. And in particular, for our procedure, that's DAP. We don't want any DAP to get from the first plate to the second plate, because if DAP comes along, those donor cells are going to start growing on our second plate, and that would be really bad. And so that's why you almost always want to do at least one restreaking or maybe two restreakings, which means going on to a second or third selective plate, just to really make sure you're not carrying over any donor cells. How are the applications different from, okay, so we have a question of how are the applications of conjugation different from transformation? Really different. So transformation, you're thinking mainly E. coli or other model organisms that have good procedures worked out. And that means having competent cells as well as the machinery or chemicals needed to get DNA into the cell quickly. Conjugation is going to be for more non-model organisms, for organisms that don't have good transformation procedures, because it takes usually longer. So for example, if you have a newly discovered organism and you're like, you know, I'm going to try this random transformation procedure that works for another organism, probably that transformation procedure doesn't work. And you're like, it doesn't work. So you can now either spend a lot of time creating a completely new transformation procedure, trying to figure out what will work for your bacteria, which can be very involved and take a long time. Or you can say, well, or I'm just going to use some the conjugation machinery that normally works in most bacteria. And you can try that route. And so a lot of people will just go that conjugation route to get things to work, because they'd rather have engineered bacteria to work with than figure out how to engineer bacteria a little bit faster. Okay. So I think that's a really big thing. There's also some bacteria that you just can't transform. Like people have worked on them for many years and they're just like, it's just super hard. It's not reliable. Conjugation works a lot more readily. So that's sort of the best there. Yeah, a couple other people have tried to also answer this, what do we mean by wash? And yeah, like they said, you're going to be centrifuging stuff, removing the supernatant or the buffers, and then taking the cells, resuspending them, and moving them forward. Can conjugation lead to mutation? The conjugation itself should not lead to mutation, but like any process inside of cells, there will always be mutations, right? So whenever you replicate DNA, there's just a chance to get a mutation, and that also applies to conjugation. I have not noticed a higher or lower level of mutation. So somebody asked, why do you need a sequence? Why do you need a 16S sequence to test, the Q&A keeps moving as people are doing stuff there. Why 16S sequencing to test the strain modification? So yeah, so let me back up there a little bit. 16S sequencing means sequencing the ribosomal. So you have ribosomes in bacteria, right? Ribosomes in all of us, but you have ribosomes in bacteria. And one of the standard ways to figure out what organism you're working with is to look at the ribosomal DNA. So basically the DNA that encodes the ribosome. If you sequence that, that is just sort of a standard benchmark that people use to tell you what the organism is you're working with. And the part of the ribosome that people like to look at the most is the 16S sequence, okay? And so the idea there is by looking at the 16S sequence for your ultimate modified bacteria, that can confirm for you if the cells you're working with are the recipient cells or if they were cells from, let's say, the donor cells. And that can be important if your phenotype is similar between recipient and donor. So if you are working with a donor cell like E. coli, which is what most people work with, and your recipient looks phenotypically very similar to E. coli, you probably need to do that 16S sequencing. If you're working with something that looks really different though, maybe you don't, right? But I would still recommend it because it might tell you if you have some donor cell contamination. So that's the goal. Best practices basically. Other questions, folks, related to conjugation? Some good questions. So, yeah. And honestly, I've only done conjugation a few times myself. My students do it all the time because of the bacteria they work with, but I myself have only done it a few times. But I think that's a good overview of transforming stuff. Okay. All right. So looks like we're good. And then I think we answered wash as well. I'm just going through the questions here. Great. Okay. So, all right, great. Thank you guys. Again, if you have more questions, feel free to send an email or reach out to me. Happy to answer. For the last part of our webinar day, we're going to go into sequencing here. Let me go ahead and move us forward to sequencing. And so we're going to talk about DNA sequencing here, and we're actually going to also do a live demonstration looking at it a little bit. I'll get online and do some stuff there with you. So part 3, DNA sequencing. Okay, for those, oh, 1 second, guys, there's a hangover Q&A question here. Let me just take a quick look. Donor and recipient cells are not the same species. Correct. Absolutely, yes. So somebody asked, can we have E. coli and S. aureus? Absolutely. That is the general idea. You're going from E. coli as a donor cell, and your recipient cell is a very different organism. That's almost always the case. Okay. Yeah. And it is generally not the same bacteria. It's almost always not the same bacteria. Okay, great. So good question from somebody there about donor versus recipient cells and the fact that they're different organisms and not just different cells.